Lastly, I did some in vitro tests to investigate the bio biological performance of the auxiliary cube. First, I infilled Gelma hydrogel, encapsulating the porcine adipose stem cells. And I also confirmed that the infilled Gelma hydrogel didn't infiltrate through the single strut. And I did some in vitro test after infilling the ASCs into the auxiliary cube. And when we see the viability of the ASCs in the auxiliary cube showed much higher viability than the control group. The control group means that the hydrogel only groups without the auxiliary cube. Also, when we measure the hypoxic cells percentage, the auxiliary, as the size of the auxiliary cube increases, the hypoxic percentage increased, but is still less than 20%. But interestingly, when we use 0.5 ml Gelma without the auxiliary cube, it showed the highest hypoxic percentage, around 40%. I'm sorry. And I also measured the cumulative VEGF that ASCs secreted inside the auxiliary cube. So ASC within the auxiliary cube secreted the VEGF much more than the control groups of the Gelma only without the hydrogel. So that means that when we use the auxiliary cube for the ASC culture, the auxiliary cube can maintain or preserve the functionality and viability of the ASCs.